Conceptual Academia. My name is Sain Dutt. I'm your biology educator for class 10th and 9th ICSE board. In this series, I have completed two videos of the chapter Basics of Biology for class 9th ICSE, which have been uploaded in this very YouTube channel. Now I'm going to cover the Basics of Biology chapter for class 10 ICSE board. Okay, so you must have noticed that we are somewhat similar to our parents. हम लोग अपने मम्मी या फिर पापा से थोड़े बहुत सिमिलैरिटीज शो करते होंगे, थोड़े बहुत सिमिलर होंगे. We will not be the exact same. The reason of which we have studied in class 9 basics of biology, that there is variable recombination of gametes from both our parental generation, that is from our mother and our father. But yet we somewhat look like our parents. कुछ हम लोगों में पापा या फिर मम्मी के या फिर उनके पेरेंटल जनरेशन के कैरेक्टरिस्टिक फीचर्स देखने को मिलते हैं. तो फिर ऐसा क्यों है? So, welcome to genetics, the first portion of basics of biology class 10th ICSE. So let's begin with the chapter, students. Okay, so in this portion, we'll be dealing with genetics. But before understanding what genetics actually is, we'll have to understand what we are trying to speak about. What is actually genetics? We'll have to understand by understanding and going back to cellular biology or cytology. When we were discussing the structure of a cell, we were discussing that there is a structure known as nucleus inside of it. तो अगर हम लोग न्यूक्लियस का स्ट्रक्चर को बाहर ड्रॉ करें थोड़े मतलब एक्सक्लूसिवली, तो फिर वो कैसा दिखेगा? It will look something like this. I'm drawing a very rough structure, students, so that you guys can understand it. Okay, so there will be another structure present inside of it, which is the nucleolus. Certain fluid matter will be present inside of it, which will be the nucleoplasm and chromatin fibers as well. Okay, so now let's label it. The external portion of the nuclear wall will be known as the outer membrane. The internal portion will be known as the inner membrane of the nucleus. These two will comprise of the nuclear wall or the nuclear membrane. Okay, so the gaps between the nuclear membrane is known as NPC or the nuclear pore complex. From the nuclear pore complex, several components will go in or out of the nucleus. Then there is this control center of the nucleus, which is the nucleolus. And there will be certain thread-like structures known as chromatin fibers. Remember, students, whenever you're trying to draw a structure in biology, do not forget to give the caption to it. So here, in this case, it is a structure of nucleus. Okay, so coming back to the main point, chromatin fibers, these things are essential. So let's take another slide. So what are these chromatin fibers? These are thread-like materials which will form chromosomes upon condensation. How do they condense? They condense before the cell divides, before cellular division, and it condenses with the help of scaffolding or histone protein. To form something which is known as chromosome. Now coming to the name chromosome, what does it actually mean? Chromo, the word refers to color or colored, and zomos or zomas will mean bodies. So chromosomes refers to colored bodies. Why do we refer chromosomes as colored bodies? Because when we try to dye or provide some kind of pigmentation in a cell to view it under a light microscope or say whatever microscope, we can see that these chromosomes will take up deeper color. Therefore, these are known as colored bodies since they are highly condensed. And if something is highly condensed, it is meant to take more color than the other cellular compartments and the other cellular organelles. Therefore, these chromosomes are known as colored bodies. How do they actually look like? Let's make a rough structure and try to understand. Okay, so these two lower portions of the chromosomes are known as the Q arm of the chromosome, whereas the upper portions are known as the P arm of the chromosome. The center portion is known as the centromere, whereas these two ring-like structures present in both sides of the centromere is known as the kinetochore. Then there is an end portion on the Q arm of the chromosome, which is known as the telomere. And there are some antenna-like portions on the top of the P arm, which are known as the satellites. So this is a rough diagram of how a chromosome actually looks like. Now, there are certain things which are known as genes, which are present in the chromosomes, which are transformed or transmitted from the parental to the filial generation, and that is the very reason why we would, to certain extent, look or inhibit the characteristics of our parental generations, or that of our mother, our father, or the generations before that. So, what is genetics? Genetics is a study of flow of gene from parental to filial generation. पेरेंटल जनरेशन यानी कि हमारे पहले वाले जनरेशन और फिलियल यानी कि नेक्स्ट जनरेशन. ओके, सो हम लोग जेनेटिक्स पढ़ने से पहले देयर आर सर्टेन टर्म्स दैट वी नीड टू नो अबाउट. तो वो क्या टर्म्स है? वो हो सकते हैं कोई ट्रेट या फिर अलील. So what are these traits or alleles? So alleles can be of a single type, such as you can say a hair color that can be either brown or black or golden. These can be the different alleles, whereas the hair color is actually the trait or the character. Okay, so these are the distinct terms that you'll have to keep in mind before studying genetics. Now, before starting genetics, we'll have to pay our respect to the man who laid the foundation of the subject, who was none other than Gregor Johann Mendel. And therefore, he is also regarded as the father of genetics. So this question often comes in exam, who is the father of genetics? So the answer would be Gregor Johann Mendel. He was not actually a biologist, to your surprise. He was actually a mathematician. But in his leisure, he worked on a certain plant, जो कि मटर के पौधे होते हैं, that is scientifically known as Pisum sativum. And he led the foundation of a biological discipline that we now today known as the genetics. So Pisum sativum या फिर pea plant was the plant that he chose for his experiment. What was the point of choosing the pea plant? This also had a significance in the understanding of his genetics. So what was the significance of Pisum sativum or choosing this very particular plant? First, the lifespan of the plant is short. And what is the lifespan? Lifespan of an organism refers to the point where an organism is born to the natural death of an organism. Birth, juvenile phase, phase of maturity, or phase of reproduction, where it is capable of producing another offspring, then senescence or old age, followed by death. So, this is the natural sequence of events that will happen from birth to death. So, this time period will be variable in different organisms. But the time period from birth to natural death of a certain organism, which is approximately 80 to 100 years in case of humans, approximately 14 years in case of uh, other birds like, say, crow, but around 140 years in case of parrots. These are examples of certain lifespans. All right. So the period from birth to natural death of an organism is known as the lifespan. So lifespan of the pea plant is short. So what is the benefit? If a researcher or a scientist were to study a human, then and he was to be studied genetics on that very particular individual, then in his lifespan, he would not be able to see the filial generations, say 10 filial generations of that very particular organism that he's choosing to study, say for example a human. But if he was to take another organism such as the pea plant, which had a relatively very shorter lifespan, then he would be able to study a larger number of generations, and his postulates will be more accurate, and he would be able to make the statements of genetics by looking at more amount of data. So by choosing the Pisum sativum plant, Gregor Johann Mendel was able to notice and study a large amount of filial generation of the particular pea plant. Then what were the other benefits? Pea plant can show both self and cross pollination. So this is another benefit, so that it can also survive in different conditions, because it can either reproduce using self pollination or cross pollination. Pollination is the process where the pollen grains of a certain plant species will be transferred from anther of the male part, or androecium of a flower, to the stigma of female part of a flower, which will be the gynoecium. So this can either be of self or cross pollination, and the pea plant or Pisum sativum is was showing both the types of pollination, that was self and cross pollination. All right. So the third reason of choosing the pea plant will be that pea plant shows contrasting characters. The way how certain people perceive colors is different. तो फिर हम लोग अगर ऐसे कहें कि से this is a color, a particular color, this is another particular color, and this is another particular color. So there might be some individuals who would not be able to differentiate between all these colors. But if you give them, say for example, black and white, so they will clearly be able to distinguish between these two colors, because these colors are contrasting and there is no match between these two colors. So there is no medium in between. It is either tall or short. So you can easily differentiate that this is a tall plant and this is a short plant. So there is no in between or intermediate. So pea plant always shows contrasting characters. This was another upper hand of Mendel, because he chose this plant for his study and reference. The other benefit of choosing the pea plant was that it doesn't show linkage or linked genes, so that intermediate properties would have been formed. So these were the benefits of choosing the Pisum sativum plant or the pea plant for Mendel for a study of genetics. But to your surprise, Mendel's work was unrecognized for 16 years even after his death. So, what were the reasons why Mendel did such a great job, but his work was unrecognized? It went unrecognized for 16 years even after his death. Why? There were certain reasons of the failure of Mendel's postulates or theories of genetics, Mendelian genetics, even for a longer duration of time, which is that Mendel was not a biologist. He was actually a mathematician. So he couldn't really earn that much of respect in the biological scientific community. The second reason could be, these are all probable reasons by the way. The second reasons could be Mendel couldn't publish his work in esteemed international journals. And the third reason would be, he said factors instead of the term genes. So we were not really understanding what he was trying to say. So these are the probable causes or the reasons why Mendel's work went unrecognized even 16 years after his death. But after his death, after 16 years of that, three scientists rediscovered Mendel's work, and that we today study as Mendelian genetics. There are certain principles and postulates as given by Mendel, such as the laws of dominance, the law of independent assortment, and the law of segregation, which are the very basic of genetics. It is these are the very important postulates as given by Mendel and are known as the laws of Mendel's. And these lays a foundation for the understanding of genetics. All right. So we'll be covering that portion in next part of our video, where we'll be studying Mendelian genetics, some important examples and theories of non-Mendelian genetics, where Mendelian genetics is not actually found. But before that, today we'll be studying certain other things, such as what a cross is, what is selfing, what is a pure line. Like we studied the two important words, alleles and traits, which lays the foundation of understanding genetics before delving deeper into it. So, what is selfing? So, allele and trait were two words. Selfing is another important word. So, selfing is actually a cross between two organisms belonging to same generation, through which pure line can be observed or a particular genetic information can be stored for the next filial generations. If consecutively 5 to 10 generation members are self or crossed between the members of same generation, female and a male organisms are mated, then we will be observing a pure line. But there are certain drawbacks of it as well, such as the inbreeding depression. इनब्रीडिंग डिप्रेशन refers to the word where जहां पे एक मेल और एक फीमेल को कंसेक्यूटिवली बहुत सारे जनरेशन में अगर हम लोग मीट करा रहे हैं इन द सेम लीनिएज ऑफ फैमिली, तो फिर हम लोग कुछ कुछ डिफॉर्मिटीज देख सकते हैं उनके अंदर में. तो इनब्रीडिंग डिप्रेशन एक तरह का प्रॉब्लम है while producing the pure line by selfing, and the process where a male and a female organism are crossed is known as a cross. So here we studied what are the words selfing, pure line, and a cross, where a male and a female are crossed or mated. All right, and this was another term which is inbreeding depression. Coming to the different terms, they can be either homozygous or heterozygous. Okay, so homozygous refers to same types of alleles. So they can either be capital T or small t, where capital T refers to as a tall organism and small refers to as the short organism. There is no intermediate in case of homozygous because both the alleles are of the same type. Whereas in case of heterozygous, one allele will be dissimilar to then that of the other, and this is known as heterozygous. Homozygous can either be dominant, where there are two capital alleles or the dominant alleles, and This can either be the recessive one, where these are the two small alleles which cannot actually dominate over the other allele. So the homozygous and heterozygous terms, I hope these are also clear. So these terms lays an important in the understanding of the foundation of genetics. So we're going to be covering the different types of cross, such as monohybrid, dihybrid, and trihybrid cross. Apart from that, there are other types of cross as well, which we study in genetics, such as the back cross, chest cross, etc. We'll also be studying that, the theories and postulates of Mendelian genetics, and examples of certain non-Mendelian genetic factors, such as pleiotropy. Then there is co-dominance. Then there is incomplete dominance, etc. We are going to be studying all these in the next part of the video. So I hope whatever we taught today is very clear to you. Whatever you didn't understand, please let us know in the comment down below. Subscribe to our channel and then press the bell icon, and also press the like button and appreciate our efforts. Whatever you didn't understand, please comment it down on the comment box down below, and we'll try to resolve all your doubts on our end. And you can also provide us with your valuable insights of how we can improve the quality of teaching. But on our end, we are trying to give you best quality education, so that you do not have to go anywhere else after watching our video lectures. Also remember to take notes while you see these video lectures, and very soon we'll also be covering all these notes and sending these PDF version of these notes in a particular Telegram channel, so that it reduces your efforts. So thank you guys, and see you in the next video. Bye.